This is the research paper that we chose for our presentation. The reason we chose this particular paper was the title is very straightforward and it doesn't focus on any particular disease but just diseases in general. Another reason we chose this paper was because of the methods used involved the genetic databases that we were introduced to in our workshops. For their research, the authors investigated the influence of disease-associated mutations on post-translational modifications, and in particular, they studied the loss of modification target sites as a consequence of disease mutation. Understanding and predicting the molecular cause of disease is one of the major challenges in biology and medicine. One particular area of interest continues to be the computational analysis of disease-associated amino acid substitution. Protein post-translational modifications are reversible or irreversible chemical alterations of a protein after its translation. The focus of the study is on the phenotypic effects of the disruption of post-translationally modified sites by the single amino acid substitution events. A number of cases where mutations of the post-translational target sites were found to be directly involved with disease, and systematic studies implicating post-translational modifications in disease are now facilitated by the rapid growth of databases containing disease-associated mutations.
The data sets of post-translational modifications were collected from several public databases and the literature, and there is a table of the summary of the data sets. First human post-translational modification was created by including only those sites that were identified in human proteins, and mapping of 25 residual long fragments from other species, modification site plus 12 amino acids to the human proteins. 25 were identical to human protein. Matching between amino acid substitutions and post-translational modifications were considered. Matches where the substitutions occurred at a modification site and matches where the substitution site was in the neighbourhood of the modification site.
For the results, we are showing Figure 3 and Figure 4 and Table 3, indicating that substitutions associated with inherited disease affect the site of post-transitional modification, with frequencies higher than expected by chance. In contrast, punitively, neutral polymorphosis affects modification sites with lower than expected frequencies. To further the study, the impact of amino acid substitutions can occur near the modification sites. Gene functions of all types of post-transitional can be analysed. The genes were separated into the gene set containing disease mutation, inherited or somatic.
In a study, disease-associated and putatively neutral amino acid substitution data was analysed and found to have 4.5% of amino acid substitutions that may affect protein function through disruption of post-translational modifications.
These numbers further indicate that post-translational modifications are not the majority cause of human genetic disease. We have found 238 post-translationally modified sites in human proteins whose mutation was causative of disease. In total, 1,289 modification sites were found to be in the close proximity to the inherited disease mutations and represent candidates for further experimental verification.
Also analysed was the enrichment and depletion of amino acid substitutions for each post-transitional modification, and we found that most follow similar trends when inherited disease is compared to the neutral polymorphosis.
I feel that more research needs to be done on this subject due to this bias, which the authors also agree with, and there may also be some contamination of the polymorphisms due to some undiscovered disease mutations.
This research paper was informative, but there have been some problems with the research due to some bias. Further research is required.
Although this paper was interesting, due to the conclusions in the discussion, it is still very early days of research of post-transitional modification. Even though it would be useful to have a central database on post-transitional modifications, as the data suggests, it is the phenotypical effects that may be more dependent on the other genes than are required before deregulation of organisms, than that of a small section of deregulations caused by amino acid enrichment or depletion.